potential for both cancer prevention and therapy. As some background for curcumin is the primary compound of dietary spice turmeric, which has long been used as both a spice and a traditional medicine in the Indian subcontinent. It has shown several promising aspects, such as being a potent inhibitor of the NF-kappa B pathway, and also by activating cell cycle arrest and inducing apoptosis in various cancer cells. The sheer amount of interest in curcumin is shown by the amount of clinical trials being held worldwide currently, with over 17 right now. And it is safe up to 12 grams a day in humans, and the mechanism of growth in the absence of p53 is less defined. One main way curcumin has shown anti-carcinogenic potential is through its ability to induce apoptosis in cancer cells. Apoptosis, which means programmed cell death, is a highly regulated mechanism controlled by several signaling pathways. Cancer cells often resist apoptosis by increasing anti-apoptosis signals and decreasing pro-apoptotic ones. But curcumin has been shown to utilize both intrinsic and extrinsic pathways, intrinsically through the activation of pro-apoptotic proteins such as BAX, PUMA, NOXA, and caspase-3, while also decreasing and downregulating anti-apoptotic proteins such as BCL2 and EGFR. Here in this diagram, the arrows show upregulation by curcumin, whereas the perpendicular lines show downregulation. Another way curcumin has been shown to be a very effective anti-carcinogenic agent is by involving p53, which is a very important tumor suppressor gene, which has also gained its name guardian of the genome. And how p53 works is that certain upstream factors such as DNA damage, oncogene activation, hypoxia, and other cell stresses activate p53, and so that it can repair DNA, activate cell cycle arrest, and also apoptosis, and therefore maintaining the genomic integrity. Although curcumin has shown a lot of promise, there's also several limitations that it has faced, such as the main one is poor bioavailability, with poor absorption, rapid metabolism, rapid systemic elimination, and limited tissue distribution, greatly lowering the effectiveness of the drug. Also, curcumin is currently less potent growth suppression than most currently used drugs, with 20 micromolars or higher doses required for effective apoptosis in most cancer cells. Therefore, in order to overcome some of these limitations, synthetic and natural analogs have been studied. The compound I'm working with, FLL22, is a synthetic analog of curcumin, and preliminary data shows promise of FLL22 as an effective anti-cancer agent, with an article in 2009 by J.R. Fuchs even saying that out of the 25 compounds that he tested, FLL22 was the most potent and selective in the breast and prostate cancer cell lines. However, not much is known about FLL22 so far, and so the purpose of my study was to evaluate the potency and also the mechanism of FLL22. First, through methylene blue staining, it was found that FLL22 inhibits cell growth in the two cell lines that I work with, H460, which is a very aggressive lung cancer cell line, and also TU212, which is a head and neck cancer cell line. As you can see here, and F1, which is 1 micromolar of FLL22, it was cell growth inhibition was greater than even curcumin 20. And in TU212, 1 micromolar of FLL22 had comparable cell growth inhibition to even 5 micromolars of curcumin. So cell growth inhibition was very noticeable. Also, FLL22 was shown to inhibit colony-forming ability better than curcumin. And as you can see here in FLL22, only the non-treatment, that's what the NT stands for, the non-treatment wells had colonies that formed. But in curcumin, up to even 5 micromolars of curcumin, there were still colonies. Also, IC50, SRB assay was conducted to find the IC50 value, which is the concentration of the drug required to kill 50% of the cells. And when conducting this SRB assay, we found that H460 in H460 and in TU212, FLL22 had a much lower IC50 value than curcumin. And here, in H460 with 0.38 micromolars required to kill 50% of cells, whereas in curcumin there was 12 micromolars. And these values were calculated using an interpolation formula. In TU212, similar results with 0.46 micromolars needed, while in FLL22, and in curcumin, IC50 value was 11.32 micromolars, so significantly higher. Also, apoptosis analysis was conducted through annexing V staining with a flow cytometer used to calculate percent apoptosis in the cells. Here, it shows that FLL22 induces a much greater percentage of apoptosis than in curcumin. With in FLL22, there was 40 to 50% apoptosis, whereas in curcumin, there was very minimal. It was comparable to the no treatment. And in 48, oh yes, go back.
You also actually emphasize the dose. For curcumin, you use higher dose, and FLL22, but since your purpose is to more potent, so people may think that you are using similar dose, but the dose is also different, 5 times lower. That's you should emphasize here.
All right. Also, in the second time point, in 48 hours, there was even higher apoptosis in FLL22, with 50 to 60% apoptosis in 3 to 6 micromolars. Whereas in curcumin, even 25 micromolars had much lower percentage of apoptosis. In TU212 cells, there was less of a difference, especially in the 24-hour time point, with FLL22 and curcumin having similar percent apoptosis. However, in 48 hours, a greater difference was shown with, if you see in the 5 micromolar, the same concentration, curcumin had around 7% apoptosis, whereas FLL22 had 63. Lastly, Western blot analyses was conducted to see protein expression. And the first one, PARP cleavage, is a further indication of apoptosis because when PARP is cleaved, you can see here that in FLL22, there are 2 lines, which means that PARP was cleaved, indicating apoptosis. But in the no treatment in the curcumin, there was only 1 line, which means that PARP was not cleaved. Also, P53 expression was highly noticeable with greater protein expression in FLL22. However, related proteins, BAX and PUMA, did not show expression, so there's probably an indication of a different mechanism. Also, EGFR and phospho-S6 kinase, which are 2 proteins that promote cell growth, were downregulated or inhibited in FLL22. So, in conclusion, FLL22 had more potent cell growth inhibition than by curcumin. There was also indication that FLL22 induces higher cytotoxicity by the lower IC50 values, inhibited colony-forming ability, and induced apoptosis while expressing P53 and downregulating EGFR and phospho-S6 kinase. So for the future direction of the study, we will evaluate and compare the potency of this drug in perhaps different cell lines, and especially more head and neck and lung cancer cell lines. Ascertain selectivity by treating normal non-cancerous cell lines with FLL22. Determining further the critical pathways of FLL22, including if it induces DNA damage, and also testing the effectiveness in vivo in animal models. And I would like to thank Dr. Rahul Amin and Dr. Aminur Rahman for all their help. Dr. Amin is my mentor, and Dr. Dong Shin, he's the principal investigator of our lab. Dr. Mary Jo Lekowitz and the Winship Cancer Institute for allowing me to intern here this summer. And also Dr. Cheryl Kirks and Ms. Kathy Banks for encouraging me to apply to this program. So thank you.